Hi everybody, this is Mrs. Bright. I hope that you're all staying healthy and doing well. The last 3 assignments that have been posted in Schoology focused on the concept of protein synthesis and specifically the first step, transcription. This week you will be taking the final transcription assessment before we move on to the second step of protein synthesis called translation. This video serves as a review of all the materials that have been in the last 3 assignments. I encourage you to take notes. You can find a copy of this diagram in this current Schoology folder. Before we begin going through this diagram, I wanted to spend a moment talking about an analogy that I use with my students called cookie synthesis. So in my family, we have a cookie that we serve at most holidays and special occasions. The original recipe comes from my grandmother, and she keeps this recipe in her recipe box in her home in Florida. When I want to make these cookies, I have to call my grandmother up and I have to ask her for the ingredients and the instructions, and I write that down as my copy. I then use those recipes to help me put together the cookie that I'll serve at special occasions. Now, it's a good thing that my grandmother holds on to this original cookie recipe, because sometimes it has that my cookie recipe has been lost, or I have destroyed the recipe by spilling ingredients on them. So, what does this analogy have to do with protein synthesis? Well, this overall process is very similar to how our body goes through the process of building proteins. Just like the original cookie recipe, your cell has an original recipe for making all of your proteins or all of your physical traits. That original recipe is what we call DNA. And specifically, the parts of the DNA that give us the instructions are called genes. Now, we don't want the DNA to get damaged or to get lost. So just like in my case, we have to make a copy. The copy that we make is called mRNA, our messenger RNA. And that mRNA strand has all of the instructions that we need in order to put together the amino acids to build our proteins. Our proteins then ultimately become our physical traits. Now, this first step of writing down those instructions we call transcription. And when we take those instructions and we build our protein, that process is called translation. Altogether, this whole thing is called protein synthesis. Now, if we go and look at protein synthesis on our diagram right here, I want you guys to imagine that this whole sheet of paper represents the inside of the cell, or the cytoplasm. In this particular piece where we see this word nuclear membrane, it's a dividing line that separates the cytoplasm from the internal parts right here, which we call the nucleus. Besides the fact that it says nuclear membrane, I can also tell that this is the nucleus because I find this molecule inside of it. That molecule has 2 strands that are twisted around each other that look like a twisted ladder, or it's referred to as a double helix. And it also has the base thymine in it. This is a DNA molecule. Now, I want to make a copy of this DNA molecule because, again, I don't want it to leave the nucleus. If it leaves the nucleus and it goes in the cytoplasm, there's a chance that it could get damaged or it could get lost. So, to make that copy, I'm going to use right here what we call RNA polymerase. The RNA polymerase is going to travel along the gene and it's going to read it one nitrogen base at a time and pair it according to RNA base pairing rules. So again, RNA base pairing rules, we will have G pairing with C, just like it does in DNA. And this time, our adenine base, instead of pairing with thymine, it's going to pair with a uracil base. So as I read each nitrogen base over here on our gene, I'm going to pair it with the correct RNA nucleotide. So if I have an A, I'll pair it with a U. If I have a C, I'll pair it with a G. Here's another C, so it should get paired with a G. A to U, A to U, another A, another U, finally another A, and another U. We hit C, so it'll get paired with the G, C will get paired with the G, and then right here we have a G, so we'll pair it with a C. And this will continue all the way through that gene until it reaches a point where it says to stop. This piece that we just built is the copy, or the mRNA. It was constructed by this RNA polymerase reading the nitrogen bases on the gene and pairing it according to RNA base pairing rules. Once the mRNA is fully constructed, it's free to leave the nucleus. And that's what we see right here in the diagram. This piece of RNA, mRNA, is leaving the nucleus. The process that we just described is referred to as transcription. I hope this video helps. If you guys have any questions, please make sure that you post them in the discussion board. We will be reviewing these posts in the discussion board and answering back, and hopefully you will be able to use this information to do well on your assessment. Have a good day.